What is actually neuromuscular junction is? This is something when two persons are talking to each other, right? And one is only listening and one is only speaking, like wife and husband. Wife is only speaking and husband is only bechara, like that. But sometime the second person replied but in angry mood, like that. This muscle will reply neurons but in other way, right? And the neuron is always talking to muscles. This is the story of neuromuscular junction. Like for example, we have spinal cord. Spinal cord. First, we have to discuss general structure, right? This is the posterior side of spinal cord and this is anterior. From this anterior horn of spinal cord, from here, the neurons are arises, right? And these neurons are called motor neurons. Like for example, this is the axon terminal. Neuron have how many parts? This is generally neuron. This is cell body, axon and axon terminals. Axon terminals. This is actually axon terminal. Lala kita kada sabi no ay hasan sta laga wande. Dita to bar da kula derni zekiri gotta. Kusa warta be huwa. Tapos mana wali nake. This is the axon terminal of neuron, motor neuron. It is also known as efferent neuron and it is also known as alpha motor neuron, right? Here the muscles is present. For example, this is muscle, right? But this neuron is always speaking, right? And this muscle will always listening. These are the ears of muscle, right? This between this gap between the muscle and neuron is called synaptic cleft. Synaptic cleft. This area is called synaptic cleft, right? This thing is axon terminal. Axon terminal, and this is actually muscle. But here the sarcolemma, which is directly communicating with this neuron, is called the end plate. This is called end motor end plate, right? This sarcolemma is called motor end plate. This thing, all the sarcolemma have these depressions. For example, here another sarcolemma, sarcomere, that have also this depression, right? And this is called, what is this? T-tubule, or it is called transverse tubule, right? On each side of this transverse tubule we have a structure called sarcoplasmic reticulum. Like, we will discuss the pathways in detail, but first this necessary the terminologies, right? This is sarcoplasmic reticulum, and this is also sarcoplasmic reticulum. These three things are collectively called triad. Triad, right? These, and the whole function is almost taking here, right? For example, he is talking to this muscle that contract. Which thing is responsible for this contraction is stored in sarcoplasmic reticulum, which is called calcium. This calcium will come out and will contract the muscle and will again go to this sarcoplasmic reticulum and stored again, waiting for other contraction, right? So we'll start. The next thing in GIT, for example, we have stomach and intestine, etc. We are eating nutrients, the vitamins, amino acids, lipids, carbohydrates. Among all the nutrients, we have one thing like vitamins called choline. Choline, right? And this choline is absorbed to blood, right? And when this blood is circulating in body, it is also passing from these neurons, right? For example, here is interstitial fluids, and this is that neuron which innervating this muscle, right? So from here it have choline. This choline will absorb to this neuron, right? We will discuss in just a few seconds, right? And also every cell have which thing? Mitochondria. Every cell have mitochondria. Like every cell, this neuron have also mitochondria, right? And this mitochondria is responsible for Krebs cycle, right? So in Krebs cycle, the first when glycolysis is entering to Krebs cycle, mean mitochondria. Here is one reaction which is called link reaction, which links the glycolysis with Krebs cycle. The main product of that reaction is acetyl coenzyme A, which is also known as acetate, right? So from mitochondria, acetate is present here, acetate. So this acetate will come out to this area, acetate, right? And this choline from nutrition, these both will react and will form acetyl choline, right? Next, then we will talk that how these acetyl choline comes out from this neuron terminal and how it interacts with this end plate, right? And what are the conditions or diseases related to these acetylcholine and muscles plates or neurons, right? So let's start. From which horn the motor neuron is arising? From ventral horn. Right? For example, I have muscle here. For example, I am taking this bicep muscle or tricep muscle. I can this bicep, and this is innervated by the spinal cord. From ventral horn, these muscles are innervated by motor neurons. We are taking this muscle and that neuron, right? For example, this is my muscle. This is the end plate of muscles, right? And from here, this is, for example, this is spinal cord. This is spinal cord, and from subspinal cord, from ventral horn, this motor neuron is arising, right? And this neuron goes and forms synapse with this muscle. These are myelinated neurons. These are myelinated neurons, right? Mean that this neuron have myelin sheets. When it have myelin sheet, the transmission of nerve impulse is faster or slower? Faster. It will be fast as compared to non-myelinated neurons. And there is one reason behind this, because it's jumping, solitary movements, right? And here is no barrier to stop that or to slow down that transmission impulse, right? We will discuss that. But here, when this spinal cord orders or gives stimulus to this neuron, right, in the form of depolarization. The resting membrane potential of neuron is minus 70 millivolts, and the resting membrane potential of muscle is minus 90 millivolts. Here is difference, right? For example, generally when this neuron is getting depolarized, what's mean by depolarization? The sodium will go inside and the cell will gaining the positive charge, right? For example, this neuron have channels called sodium channels, and from by these sodium channels it takes the sodium in, right? Here it gets depolarized. Depolarization mean that, for example, the resting membrane potential of this neuron is, this is resting membrane potential is minus 70 millivolts, right? When these positive charges go inside this neuron, the electronegativity will be decreasing, and this minus 70 will goes to minus 60, minus 65, minus 50, minus 55. When it reaches to minus 55 millivolts, this is called threshold potential. Threshold potential. And by this threshold potential, it will activate very specific channels here called calcium channels. These are calcium channels, and calcium will rush into neurons, right? But here this neuron have a specific type of neurotransmitters. What is neurotransmitter? Anyone? What is neurotransmitter? Neurotransmitters. These are a specific type of proteins which are responsible for the transmission of impulses in neurons, from neuron to neuron, and from neurons to muscles, right? These are the specific type of protein formed in cell bodies. These neurotransmitters are formed in cell bodies, but these vesicles are synthesized here. Am I right? Am I right? No. The neurotransmitters are synthesizing here. I have already told you just a few seconds back. Think. Choline and acetate forms the neurotransmitter here. The cell body is only responsible for formation of these vesicles. Mean that when from cell bodies the vesicles are coming here, they are empty bags. right? You are sleeping. You all are sleeping. We are taking one cell body and axon, and we'll see that how these vesicles are transporting, right? For example, this is my axon, right? And I am taking here the cell body. Here is cell body, and these are dendrites, right? This is neuron, right? This cell body have, this is actually the cell body where all the machinery of neuron is present, like all cells, right? So here we have a nucleus. We have nucleus. This is nucleus, and nucleus, which thing is present? DNA. Here DNA is present. For example, this is my DNA. DNA, right? Some specific genes are present on this DNA which are responsible for the formation of these vesicles, right? So it will form messenger RNA. For example, this is messenger RNA, right? From messenger RNA leaving this nucleus by nuclear pores, and this messenger RNA will comes out, right? This step is called transcription. Now translation is, for example, this is ribosomes, right? And by this ribosome, this DNA has been scanned, and which thing is formed? Vesicles. These are vesicles. These are very special type of proteins, very special type, because some proteins are attaching to these vesicles, right? And we will discuss that what are specifically that proteins are, but commonly it is called V snares. These proteins are called V snares proteins, right? And it also have some specific channels here, right? This channel is called acetylcholine transporter, acetylcholine channel, right? Acetylcholine channel. If this vesicle is formed for serotonin, it must have serotonin channel. If it is formed for epinephrine, it must have epinephrine transporter, right? And one protein, one channel is here. It is called proton pump hydrogen, right? But now still this vesicle is empty, right? And also the mitochondria are present in cell body, because it is a cell, so it have a lot of mitochondria. And I have already told you in cytoskeleton that in middle of this axon we have intermediate tubule. For example, this is intermediate tubule, right? And two specific proteins are attached to this. One is standing here, right? And one is standing here, right? This protein is called dynin. This is called dynin, and this is called kinesin. Are you recapping these names?
Yes, sir.
Right? So this protein will go from here when these both the domains are filled and there is action potential, right? For example, I have another neuron. This is actually the dendrites, and we have the axon terminal of another neuron here, right? This whole story is also running in this neuron, right? For example, this action potential is transferred to this neuron, right? The channels here, sodium channels, will be activated, right? And this neuron is depolarizing, right? Here, by this depolarization, this neuron also have a specific channel in this axon hillock. This is axon hillock. This is also calcium channel. Calcium goes inside. By this voltage, these sodium channels will activated. For example, this neuron is depolarizing. This is now depolarized, right? And by this way, this protein, for example, here during this process, here which process is taking place? For example, we have GIT. This is GI, right? And we are eating choline. As like other vitamins, we are taking choline in our diet, right? This choline is absorbed to blood. For example, this is blood, right? And these cholines are absorbed to this blood, right? From here, it will goes to the extracellular space. Extracellular space is rich in potassium, magnesium, sodium, calcium, right? So this area is rich in sodium plus calcium. This choline goes to this extracellular spaces, choline, and from here it goes to this neuron, right? It goes to neuron. Now choline is present here, right? Choline is present here, and it needs one mitochondria from which it will take acetate, and it also need a bag in which it will store, right? So from here, this axon terminal send this kinesin, right? This kinesin is sent here to tell this dynein, please bring one vesicle and one mitochondria. This is the actual transmission, right? So here, this is called choline channel or choline receptor, right? Here, this dynein, he is taking vesicle in one hand, right? And in other hand, he takes mitochondria, right? If mitochondria is deficient here, right? And when these both the domains are filled, now this protein, according to this action potential, it is going in this axon, right? And bring these vesicle. For example, this is the vesicle and this is the mitochondria. Both are now in axon terminal, right? And as I have told you that this vesicle have acetylcholine transporters, acetylcholine receptors, and also this vesicle have proton pumps, right? From here, from Krebs cycle, for example, I have 6 carbon compound here, glycolysis, right? I have 6 carbon compound which is called glucose. This glucose undergoes glycolysis and forms 3 carbon compound which is called pyruvate, right? By pyruvate dehydrogenase enzyme, pyruvate dehydrogenase, this will goes to a mitochondria but in changed form. For example, it lost one carbon dioxide and remaining thing is acetate, right? And coenzyme A is binding to acetate. Now this is acetyl coenzyme A. From this mitochondria, this thing is present in mitochondria, actually in the mitochondrial matrix, right? From mitochondrial matrix, it gives the acetyl to this neuroterminal, axonal terminal. Here, these both the things will binds in the presence of a specific enzyme, and it will form acetylcholine, right? Acetyl and choline are binding and forms acetylcholine. Acetylcholine, these formations occur in the presence of a very specific enzyme, and this enzyme have a name, a very good name, choline acetyl, sorry, choline acetyl transferase. Choline acetyl transferases. These enzymes are working here in this reaction and forms acetylcholine. Right? Now acetylcholine is formed. Which thing is deficient? The vesicle. So for example, this vesicle is brought by the dynein and present here having the specific protein transporters. This acetylcholine, for example, this is that acetylcholine, but this acetylcholine have also this coenzyme A group. Acetylcholine have also this group, right? And this also have two parts, choline and acetyl. So acetyl coenzyme A and choline mean that it have it is just like this, right? And this is why this is necessary, because one other thing, nicotine have also the structure like the second part, choline, right? And they can act on same receptors, nicotine and acetylcholine. So the receptors present here for this acetylcholine are also the receptor for nicotine, right? And so they are called nicotinic receptors. So what is nicotinic receptor? The receptor which are sensitive to acetylcholine or nicotine are called cholinergic nicotinic receptors, and cholinergic nicotinic receptors are present here, right? And they are ligand binded or voltage gated. They will be ligand binded or voltage gated. Ligand binded, these are ligands. Acetylcholines are ligands and they will attach to that receptors and will activate it because they are sensitive to acetylcholine. Now this acetylcholine ACH, acetylcholine will goes into this vesicle, right? By which thing? By this acetylcholine transporter or channel, right? Now these acetylcholine is present in vesicle, right? Also hydrogen ions goes to here. Now this acetylcholine is moving in concentration gradient, but this hydrogen ion is going in against concentration gradient, because the hydrogen ions concentration here is very high and here low, and this hydrogen is moving inside. What it's mean? What it's mean? It's mean that this acetylcholine is regulated by this hydrogen ion. If hydrogen ion concentration is lowering here, it will stop the formation of acetylcholine. And when this hydrogen ion is increasing in exon terminal, this is the indication for synthesis of acetylcholine. So this acetylcholine formation is regulated by this hydrogen ion. For example, I have 3 hydrogen outside the vesicle, and I have 5 hydrogen inside, right? And acetylcholine is going inside. With acetylcholine, these hydrogen ion will also goes inside, right? So when here acetylcholine is decreasing, hydrogen ion also decreasing, and this is the indication that this vesicle is filling, right? Now if this acetylcholine will be transported to synaptic cleft, so the acetylcholine inside will be decreasing, and this is the indication that these hydrogen ion will again leave this vesicle, and the hydrogen ion concentration here will be increasing, and this increasing value is the indication for formation of acetylcholine, because from this high concentration it is a signal that acetylcholine is leaving this exon terminal, right? So this is the formation and vesiculation of this acetylcholine, right? Now here is one disease which is caused due to this calcium insufficiency. These are the calcium channels, right? And when these calcium channels are blocked, these calcium channels are blocked, this vesicle is now not able to release it, because these vesicle have a specific type of proteins, right? These proteins are called V snares. These are called V snares, and these are called M snares, membrane snares, or T snares. These are called terminal snares. Two proteins binding here. I am making this diagram here. For example, this is the vesicle having acetylcholine, a large amount of acetylcholine, right? And it have a specific type of proteins, right? And these are called V snare, vesicular snares, right? This protein is called synaptobremin. It is called synaptobremin. Like for example, it is called synaptobremin, right? And this protein is called synaptobremin. You have heard about the medicines pyrotegmin, neutegmin, which is used in the weakness. They are actually the analogs of these proteins, right? This is called synaptotegmins, right? And this membrane also have these proteins, right? For example, this is the vesicular membrane. This is the membrane of neuron, the terminal membrane, and it also have these proteins. Here, these are called T snares, but the specific name for this protein is, this is called snare, or it is called SNAP 25. SNAP 25, right? And this protein is called cyanotaxins, right? Cyanotaxins. So these are the proteins which are bind. What is the function of these proteins? We will discuss this contraction, but now we are discussing only the general structure and functions of this neuromuscular junction, not neuromuscular junction, only this neuron, right? What is the function of these proteins? Binding. And this is wholly solely calcium dependent. This is the reason why these channels are present here, this calcium, right? For example, I am taking this green membrane here. For example, this is the green membrane, right? And the target of acetylcholine is what? To release it into the synaptic cleft, right? Here I have a vesicle, and this vesicle have protein, like for example this is the synaptobrevin, and this is the synaptotegmin. Also here this membrane have SNAP 25 and cyanotaxins, synaptotaxins, right? What really happens when calcium goes inside here? By which thing? By this calcium channel. So this area is now calcium rich. Calcium rich. This calcium came here and binds these both proteins, right? This also have bind to calcium, right? And what happens that, for example, this is that membrane, and now this is the SNAP 25, and this is the SNAP, this red is synaptobrevin, and this synaptobrevin is now binds to this SNAP 25, right? And it contracts, right? And on this way, on this side, this is cyanotaxins, right? And it will bind to synaptotegmins. Which thing is responsible for this binding? Calcium, right? Now this vesicle is present here, and it have a lot of acetylcholine, right? What really happen that these both the membranes binds to each other, like in this way, and this process is called exocytosis, when something is getting out from the cell. And when these membranes are binds, the acetylcholine came into the synaptic cleft. For example, this is now the acetylcholine is present in synaptic cleft. But one vesicle is not enough for signal transmission, because one vesicle approximately it forms 0.4 millivolts voltage here, and it requires more than one 10 millivolts. By 10 millivolts or 30 millivolts it will get depolarized, right? So we will discuss that potentials, but this is the exocytosis, and now the acetylcholine is present here, acetylcholine, right? When this happened, after this, when this vesicle is now empty, because it gives all its acetylcholine to synaptic cleft, right? Now this membrane is, for example, it is present like this, right? It is empty now. What's really happens that this neuron is very intelligent, and he is not ordering for other vesicle because they are expensive. These vesicles are very expensive, they are very special protein, and this neuron is not leaving this protein in this membrane, right? Here one another protein came and bind here, and actually this protein is called clathrin. And I think about clathrin, in every paper this MCQ is present. Clathrin, right? This, what is the role of this clathrin? This clathrin, it again forms this vesicle from this membrane and binds this to original structure, right? And now this vesicle is empty and it will again filled by acetylcholine and will release. So clathrin are responsible for the restoring of these vesicles, right? Clathrin. So up to now we have discussed how many proteins? Dinins, tyrosines, micro intermediate tubules, right? Choline, acetate. The next thing, V snares and T snares. V snares are synaptobrevin and synaptotegmins and SNAP25 and cyanotaxins. Right? Now, what really happens here? Here are, for example, this is the muscle, right? And you have already discussed the cell membrane. Cell membrane have a lot of receptors and channels. Here the receptors are present and they are ligand binded channels. This channel have a very beautiful ears and listening from this neuron. Only listening. From only listening, I am trying to clear that this is one-way transmission. This is one-way transmission. Only the transmissions are coming from here to muscles, not from muscles to neurons, right? If this muscle want to respond, it will be abnormal, and the condition is called myasthenia gravis syndrome. Myasthenia gravis syndrome, and the second syndrome is Eaton Lambert myasthenic syndrome, right? We will discuss this. This is the channel and this is called nicotinic receptor, right? Nicotinic receptor. Why it is called nicotinic receptor? Because this acetylcholine can bind here. For example, this is the domain for acetylcholine. So acetylcholine can bind here and can activate this receptor, right? But like this, nicotine can also bind here. So these are called cholinergic nicotinic receptors. Cholinergic receptor, this is a type of cholinergic receptor. Cholinergic receptor have two types, the NM receptors and NN receptors. These are NM receptors. NM, this is the receptors which are present in neuromuscular junction. One is present here, one type is present here, and these are called N receptor because it is present between the neurons and neurons. N receptors and M receptors, but specifically it is called nicotinic receptors or cholinergic nicotinic receptors, right? From here it will bind to this. The structure of this channel is, it have 5 proteins. For example, this is this channel, right? It have this green protein, right? It have this red protein, also it have this black protein, right? And behind it have these, one is blue protein present here, blue, and one another present and is red, and this is also red, right? This red is called alpha 1, this is called alpha 2, this blue is called beta, this black is called delta, and this is called gamma, right? So this receptor is also known as alpha 2, beta, delta and gamma receptors. Understand? This is also known as alpha 2, beta, delta, gamma receptor, because it have two alpha proteins, one beta, one delta, one gamma protein, and the this receptor have 5 units of protein, and it form this channel, right? And it is sensitive to ligand, and here the ligand is acetylcholine. This is ligand. So this is ligand binded ions channel. When this acetylcholine is binds here, the sodium will goes inside, but potassium will leave outside, right? Here one another thing I want to explain, that these receptors are like co-transporters. For example, this is this transporter. When sodium will go inside, potassium must be, because this muscle is trying for homeostasis, right? And when it is become positive, more positive inside, it is trying to keep it negative, because in resting state it is in negative state. So these potassiums are leaving for this property, that this muscles wants to restore this negativity, resting membrane potential, right? But one thing here, why it gets depolarized? If sodiums are coming in and potassium are coming out, why then it get depolarized? Here is one reason. When 3 sodiums goes inside, 2 potassium will goes outside, right? And so it have one positive charge more than outside, so this muscle gets depolarized, right? So here the sodium will goes inside and the potassium will goes outside, and it will get depolarized. But this depolarization is not enough for this muscle, because here thousands and millions of vesicles are releasing these acetylcholines, right? If, for example, I have nicotinic receptor here, I have nicotinic receptor here, and I have nicotinic receptor here. For example, this is 3000 nicotinic receptors, and due to some reason, these, like for example, if we are taking medicines like Resamical, they are umbrellas, these nicotinic receptors, right? And acetylcholines are not allowed to bind these receptors, right? If this happens and only one channel is remaining here for activation, and for example this is acetylcholine, and this channel activated and it start depolarization, it is only responsible or able to form 0.4 millivolt depolarization, which is very less, very less, and this is not enough to depolarize these muscle, right? So the point I am trying to clarify, that for example 0.4 here, 0.4 here, 0.4 here, all the summation, these potentials become 8, right, and gets depolarize this muscle, right? Now for example, this is the muscle and depolarization starts from here, right? This is ligand binded, so it is slow. as compared to voltage gated. It gives these potential to sodium channels present here. These are sodium, a lot of sodium channels are present here, and these sodium channels have one activation gate and one inactivation gate. We will discuss the structures, but by this depolarization, for example, this is the graph of muscle, and this is the resting membrane potential, which is minus 90 millivolt, minus 70 is in neurons, right? Now by taking the sodium inside, this potential goes up. For example, minus 80, minus 70, minus 60, then minus 55, minus 55, and minus 55 is called threshold potential, and this potential is responsible for activation of these channels, because these are voltage gated channels. These sodium channels are voltage gated, and the voltage came from here. For example, minus 90, minus 80, minus 70, minus 60, and minus 55. By minus 55, these channels become active, and sodium goes inside, and the cell gets highly depolarized, and this depolarization will transfer this depolarization to sarcoplasmic reticulum. And after now we will discuss that how this transmission goes here, and which proteins are responsible for releasing the calcium from, because our target is releasing of calcium, right? Some called sequestrins proteins are responsible here, and others are, I think, diapyridones. Diapyridones are also responsible for this, right? We will discuss. But this is the formation of acetylcholine. This is the transmission in neurons, and how this transmission is transferred from neurons to muscles and synaptic cliff. We will discuss this in detail now, right? So push back, Anna.